This is a production of Cornell University.
So also thank you for giving me this opportunity to present our work on watermelon. Okay. So here is the outline of my talk today. First, I will provide a little bit of background for watermelon, although most of people already know, And then I talk about the generation of high-quality reference genomes of watermelon. And then I will talk about the genome resequence of diverse representative watermelon accessions. And then I will talk about the GWAS and the QTL mapping of important watermelon traits using the resource we generated. And then at the end of my talk, I will spend like a couple minutes to introduce a new project. It's called QQCAP. So as most of you may already know, that watermelon is a member of a cucurbita family, which includes melon, cucumber, watermelon, and pumpkins. Several very important fruit crops. And this is a fruit map. and you can see watermelon is a worldwide fruit crop. You can see watermelon everywhere in the world. And watermelon is also an economical crop. And so in 2000, this is the data I got from FAO 2012. The whole vegetable is 57 million hectares was grown. About 6% is for watermelon. And in China, even more, you got 7%. China is the leading producer of watermelon. And you can see the production in the world is about 105 million tons annually. And China produces about 7% of the watermelon in the world. And also the consumption, per capita consumption is about 3.5 times in China than the average of the world. So watermelon is also the most popular fruit worldwide based on the production. And you can see watermelon ranked #1 on the production. So tomato is not counted here. Tomato is, if you count tomato as a fruit, then tomato is the #1. Tomato is about 160 million. But most people treat tomato as vegetables, not fruit. So you can see the several popular fruits: watermelon, banana, apple, oranges, grapes. Watermelon, the production is ranked #1. So watermelon is also a very nutritional crop, and this is a nutrition fact. And also, the watermelon contains very high levels of lycopene than any other fresh fruit or vegetable. It has contains like 15 to 20 micrograms per 2-cup serving. That's very high. And the watermelon also contains this citrulline. So the name of citrulline was derived from the Latin name of watermelon. So the Latin name of watermelon is Citrullus lanatus. So that citrulline was derived from the citrullus. So this is a very important amino acid that maintains cardiovascular function, and also there's a report says that this amino acid have some Viagra-like effects. But it's certainly good to your heart, to your health. So watermelon is also emerged as a model system for non-climacteric fruit quality development. When people study like sugar accumulation, fruit ripening, and rapid fruit expansion, watermelon become a very good system. And also the cucurbita species like watermelon, melon, and cucumber, they also as a model system for flower sex determinations. So they have 3 different flower types: so male flower, female flower, and a bisexual flower. And based on combination of these different flower types, one plant could have different flowers. So you have different flower types plant, like this monoecious that only have male and female flowers in one plant. And monoecious is male and bisexual flowers. So these are two major flower types in watermelon. But in cucumber, you have all kinds of these 7 different flower types. But watermelon now has, the cultivated watermelon have very narrow genetic diversity. Although their phenotype diversity is very large, but their genetic diversity is very, very narrow. That's causing the bottleneck for breeding. And also the watermelon has a complicated quantitative traits. I think this same as other crops, like fruit size, sugar content, shape, fruit shape, the flavor, and maturity, shelf life, uniformity, growth vigor, etc. And also watermelon is sensitive to quite a lot of diseases. Like this is the most important disease in watermelon. It's a Fusarium wilt. And also it's there's a lot of pests in watermelon, like aphids and also root knot, nematode. So because of these problems, the narrow genetic diversity, all kinds of different complicated traits, and we need modern tools, such as the genomic tools. That's what the focus of the talk today. That needed to be developed for watermelon to facilitate its breeding and research. So next I will talk about the reference sequence we generate for watermelon. So far we have generated 3 reference genomes in like last 7 or 8 years. The first one we generate, so in 2008, we formed an international watermelon genome initiative. And we are, the goal of this project, the watermelon genome project, one of the goals is to generate a high quality draft genome of watermelon cultivar 97113, is this cultivar. So this is an East Asia type. It has early maturity and high fruit quality. And we also have these additional objectives. One is the resequencing of the watermelon accessions that I'm going to talk today also. And also we generate a lot of transcriptome data involved in different interesting biological process, but this I will not talk this today. So watermelon is, the genome size is about 425 million base pairs. And it's highly homozygous, and it can be easily inbred. So even in 2008, it's not that difficult to do genome assembly. So we did a genome assembly using the whole genome shotgun approach, purely Illumina sequence. And at that time, the main sequence is like 50 base pairs or 75 base pairs. Now it's like 250. But we still managed to assemble a very high quality genome. So the N50 size is 2.3 million base pairs. That's pretty high compared to all other assembled genome at that time. And we also assembled about 80%, more than 80% of the genome. So the genome size is 425, we assembled 353. So we did, at that time, we did analyze the unassembled reads. We found most of them are repeatable sequence. So that's what we expected. And we did a genome annotation. We identified about 23,000 genes. It's less than most of the crops. That tomato had 35 million. Even Arabidopsis had 35,000. Even Arabidopsis had 27,000 genes. So the reason we have much less genes, this could be one reason. So the first thing we analyzed this genome, we found there's no whole genome duplication event occurred in watermelon and also in cucumber. We didn't find any recent whole genome duplication. But in Arabidopsis, they have 2 recent whole genome duplications. And in tomato and potato, we have a recent whole genome triplication event. And all urticas have an ancient triplication. And you can see this ancient triplication also exists in watermelon and cucumber. That's expected, So without this whole genome duplication, then that's why we got much less gene numbers in cucumber. And recently we sequenced the pumpkin genome. This is also in the cucurbit family. We found a whole genome duplication. And the gene number in pumpkin is like 32,000. It's much higher than cucumber. We did some comparative genomic analysis. So our finding is that the transition from the 21 chromosome eudicots, so the eudicots has an ancient ancestor which has 21 chromosomes. So that involved a large number of fissions and fusions. So in order to reach the modern 11 chromosome structure of watermelon. And you can see this mosaics of different ancestor blocks. So during the whole genome evolution, this shows how the genome from the ancestor come to the modern watermelon. And also we identified large synteny. We found a lot of synteny between the cucurbit species, the melon and the watermelon. We also checked this citrulline metabolic pathway. And the interesting thing is that, so we compared to Arabidopsis. And this is the pathway. The only thing we found that there's a expansion in these two families. In Arabidopsis, there's only one gene members, but in citrulline, in watermelon, we have two members for these two gene families. And then we use RNA-seq data. We checked the expression pattern, and we found that three genes are downregulated during fruit development. That corresponds to the high, elevated citrulline content during fruit development, because these two gene families are involved in degradation of the citrulline. So this is a citrulline, then this degradation, although this form this circle and feedback loop. But I mean, we believe that citrulline accumulation in the maturing fruit is likely due to decreased activity. So this decreased activity of citrulline degradation. Okay, with the rapid advance of sequencing technology, so we recently sequenced another genome. So the 9713 is an East Asia ecotype. So now this one was an American ecotype. So you can clearly see the big difference of these two ecotypes. So the fruit shape is different. This one is round, this one is oblong. And then the fruit size, this one is much larger. This one is about 25 to 35 pounds, and this one is about 10 to 15 pounds. And there are also, other phenotypic difference. And the reason we sequenced this one because of a group in Charleston. So this one was developed in Charleston, South Carolina, and they are using this as a system to develop all kinds of studies. So we also using the whole genome shotgun, using the Illumina, the most recent Illumina technology. We didn't use any PacBio. And you can see the difference, after the technology evolvement, This was done in 2009, 2010. This was done in 2014. And you can see the N50 size from 2.3 million to 7.5 million increase. And also the genome we assembled. So when reads getting longer, then a lot of repeated sequences now can be assembled. So we assembled about more than 90% of the genome. So this genome probably the highest quality we have assembled so far in my lab. We got about the similar number of genes, and then we identified the single nucleotide variations between these two, between East Asia and American ecotype, and we found that the genome of these two species are highly similar. This is only the SNP rate is only about like 6 SNPs out of 10,000 base pairs. That's very, very low. And we also identified structure variations, larger insertions, deletions, inversions, or duplications. And we identified mainly deletions, we identified 271 deletions in Charleston gray, this American ecotype. And only, you can see when we talk about the wild reference genome next, you can see the big difference. So now we only have 233 kilobase deletions in the genome, and also 500 kilobase, 600 kilobase in this 9713 genome. And interestingly, we found that these deletions are enriched in this fluid weight QTL region. So we have these orange lines are fluid weight QTL. We have 5 fluid weight QTLs. So the chromosome 6 to 11 I didn't show here because they don't contain any fluid weight QTL as far as reported. So you can see the 36 of these deletions are in these 5 regions. It's highly enriched. And that's understandable because you can see this fruit, the fruit size, fruit weight are highly different. Okay, next I will talk about a wild watermelon, the reference genome. This is more interesting than the American ecotype. And we initiated this wild watermelon before that Charleston gray American ecotype, and you can see the highly the phenotypic difference of these two accessions. The wild species, there's no sugar content. You don't want to eat it because also bitter, you know. And it has white flesh, and the hard texture is very hard, and it has late maturity. It's also mature, but it's very late. But the good thing of this one is the resistance to the flucloxamine real, to the FUNRIS 012. And NASA13 is sensitive to this one. So we did genome assembly. The assembly, the quality is about the same as NASA13. So we emphasized about 2.1 million base pairs, and we assembled about 80% of the genome. And then we did a comparative genomic analysis and using molecular clock to date the speciation events between the wild and cultivated watermelon. And we found out that these two species was, the speciation event was occurred about 1.3 million years ago. So tomato and potato, the speciation event occurred about 6 million years ago. So this is much, because it's the same species, it's a much closer than tomato and potato. And we identified genomic variations, and you can see now we have 3.9 SNP identified, compared to 200,000. That's like 5% of the SNPs identified in between NASA13 and Charleston gray. So that means this genome and the wild species and the cultivated watermelon, their genome are highly different. There's a lot of difference. And you can see this almost 16,000 genes were affected by these variations. That's about 80% of the genes in the genome, 70%, maybe 70% of the genome. And we also identified 680,000 small indels, and which leading to frame shift in 247 genes. So we also identified the large structure variations, the present and absent variations, and we identified, in NASA13, we identified 6.7 million base pairs, compared to like 200 or 600 kilobase. It's much, much more. And the gene affected by this is 270, and also we identified 5.7 million present variations in this wild watermelon. It contains 223 genes. As I mentioned before, so this wild accessions, they have resistance to FUNRIS 02 and 01 and 2. And we compared this genome, and we found that the R genes, MES resistant genes, we only found that two genes that are totally unique, that only exist in this PI, and one gene was exist in this NASA13. So that means this gene are totally absent in one or another. But when we check this present and absent variations, because some genes they are not totally gone. The variation may be just cause a frame shift, maybe truncate a part of an intron. So we found several MBS-LR genes that are in this list, and also disease-resistant genes, disease-related genes like receptor-like protein kinase, and we also found some genes that relate to abiotic stress like heat shock proteins. So this may also explain the difference between these two, the phenotypic difference of these two watermelon accessions. So one is highly resistant to disease and also can grow in very harsh conditions, like desert, a high temperature. I think this stuff may relate to the flavor. I'm not sure. We also identified the inversions. We identified 6 inversions. And interestingly, we identified one interchromosome rearrangement event. And you can see this is in cultivate watermelon. This is the wild watermelon. So in cultivated watermelon, this watermelon chromosome 11 in wild, this part was rearranged into chromosome 2 in cultivated species. We did several checks to make sure it's not assembly error. We using the paired end rates or met pair rates to confirm it is a real interchromosome rearrangement. But the implication of this rearrangement in watermelon qualities or phenotypes, that's we don't know. We don't know yet. Remains unknown. Okay, so we generated the watermelon reference genome, which provide a great resource. Now with this resource available, now we can do genome resequencing. So we did genome resequencing with a wide range of watermelon accessions. So watermelon was thought to originate from Africa. So was originated from Africa, then distributed to the world. But whether it's originated from Southern Africa or Western Africa, so now it's still a debate. There's no clear evidence to support that. Most people thought it's originated from Southern Africa. But recently people argue that they may be originated from Western Africa. It's still not resolved yet. So this is the wild watermelon, but in about 4,000 years ago, the seeds as well as the paintings of watermelon. This painting of watermelon was found in Egyptian tombs that's built more than 4,000 years ago, and you can clearly see that this watermelon. I'm not sure this picture shows. So the shape, the first shape is different from the wild type. So wild type watermelon is round shape, and that means that 4,000. This one is a little bit long, right? I'm not sure whether it's that clear. So that means in 4,000 years ago, people is already started to domesticate watermelon and use watermelon as a fruit. This called a pharaoh's fruit in Egyptian. So this is a watermelon in the 17th century. So you never see watermelon now, this kind of watermelon in nowadays. So, in 17th century, the watermelon is like this. Maybe you think, oh, people living in that area is very tough, you do this kind of watermelon. So then this is a modern day sweet dessert watermelon. This kind of like the evolution history of watermelon based on the available evidence. But the problem is that the scenario of watermelon evolution still remain largely unclear. The first one is what is the ancestor of watermelon? And we know this white watermelon, there's all kinds of different white watermelon, but which one is the ancestor, the ancient ancestor of watermelon? And the second one is what is the direct progenitor of cultivated watermelon? And also, what's the domestication process? So to answer this question, I'm sure it's very hard to clearly answer those questions, but we are trying to using this genomic resequence data to answer part of these questions. And the watermelon has 4 species. So this red one is Citrus lanatus. That's the watermelon species. It also includes 3 subspecies. So this one is cultivated watermelon. One is American cultivated watermelon, also divided into American ecotype and East Asia ecotype. And then we have this semi-wild watermelon. And then we have Citroides, that's it's called wild watermelon. And then we have a C. Icohorses watermelon. That's Citrus rehami. And then this collisensis. And also this one. So this is this wild species, and this one is now the Neonos. It's hard for me to pronounce this one. It's originally called the Johannesburg cucumber, but they classified it as a citrus. And you can see this is a fruit of that one. And also the taxonomy in watermelon is also a huge debate. So this is people classify this as a 5 different species, but now people said it's probably 7 species. So they class this one as different species, not subspecies. So it's still a debate, So we did we selected 174 representative accessions from a germplasm collection of 2,000 accessions. And we did genomic resequencing. We generated a total of 910 gigabase data, and the average resequence coverage depth for each genome is about 15x. That's pretty high. So we identified 24 million SNPs, identified a lot because of those wild species. But you can see we only identified about 1 million SNPs from this cultivated species within those groups. And we identified 5 million in semi-wild, another close to 6 million from Citroides, and 7 million from collisensis. And we use this SNP to derive the genetic diversity using the pi parameter. And we found the cultivated species, the genetic diversity is less than 1 out 10 to -3. And the collisensis, the genetic diversity of collisensis is pretty high. It's 6.48. When you compare this to other crops, and you can see the genetic diversity of watermelon is extremely low, even lower than cucumber. Cucumber is the cultivated one is about 1.85. East Asia is lower, is greater than 1, And the rice, indica 5.7, Japonica 3.7. And the soybean, the cultivated one is close to 2. So this confirms the previous study that the cultivated watermelon has very, very, very narrow genetic diversity. That causes huge problem for breeding, a bottleneck for breeding. We then use this SNP to build this phylogenetic tree. And the first thing we found is that this one. We use cucumber as a root, as an outgroup. And we found this one was not clustered to a watermelon. It's clustered to cucumber. You can see these two clustered together. And as expected, we found a lot of all the cultivar species, they cluster together, then this semi-wild, then this Citroides, and this wild species, they cluster together. And then we have thought about, does this one is a real watermelon, or is a melon, or is a cucumber? So we did some analysis. The first one we did is based on the phylogenetic tree clearly shows it's not watermelon, right? Then we mapped the race to all kinds of different genomes we have. We have cucumber genome, we have watermelon genome, we have melon genome. We also have 2 pumpkin genomes. We align the resequence rates to them. We found it matches most to melon. And also we did fish, and we found the chromosome number of this one is 24. It's 12 chromosome pairs. That's same as melon. The cucumber has 7 chromosome pairs, watermelon has 11, and the melon has 12. So we conclude that this one is not a watermelon species. It should be a melon species. So then we did not use this one in the downstream analysis. So we did a population structure analysis, and the conclusion from this population structure analysis is that this semi-wild watermelon should be the direct progenitor of the cultivated watermelon. And originally people thought this colescensis should be the ancestor, the ancient ancestor of watermelon. But this population structure analysis did not support that, because you can see we didn't see any this orange block, the orange allele. We didn't see in the cultivated species. We didn't see any orange block from this from this colescensis. And we hypothesized that these 2 species, you can see this have green, blue, orange, and this was cyan or whatever. So that's have this all colors from the cultivated species. So we hypothesized that these 2 could be the ancient ancestor of watermelon, but this conclusion is not that conclusive, because we didn't sample a lot of species, but we can rule out this colescensis as an ancient ancestor. And also we found that this one, this Citroides or Lanatas, the subspecies, they could be a separate species instead of a subspecies based on this population structure analysis. You can say it's highly different from the other species. So we then identified the selective swap. We using the pi ratio between the semi-wild watermelon and the cultivar watermelon. So we identified the select swaps across the whole 11 chromosomes. And one thing that's very interesting is that we identified this select swap. You can see the huge genetic diversity reduction in this region. And we found this region corresponds to a fruit weight QTL. So fruit weight is a major trait that was selected during human domestication. Then we found this one. And also we found a lot of other interesting stuff but I will not talk about here due to the time constraint. So watermelon, it's a major problem, as I mentioned before. A major problem for watermelon is a disease. And we checked the watermelon genome. We identified 46 MBSLR genes. That represents the least number of MBSLR genes in any genomes of crops. You can see cucurbita species normally has very few, but watermelon even has less than cucumber and melon. And other like tomato, you got several hundred, but in watermelon you only have 46. That's cause problem for watermelon. So then we think about, because the wild species, they are highly resistant to watermelon, to all kinds of different diseases. And we hypothesized that maybe some genes was lost during the domestication. So we did some analysis. This analysis was not done on the 134 accessions. It was on 20 accessions we did at the first time, but the results should be similar. So we align the reads to the reference genome, and those that are aligned, we do a de novo assembly. we divided those unaligned to two different groups. The group 1 is from the cultivated watermelon. And then the group 2 includes semi-wild and wild watermelon. And you can see the group 1, we didn't find, this is the gene was missing in the watermelon genome, but we didn't find any disease genes. Then from the wild species, we de novo assembly of those unmapped reads to the wild species, and we found it's highly enriched with disease-resistant genes. And we then map these reads to confirm this. This is not a sequencing assembly error or whatever, or in the genome. We map the cultivar reads. We map the reads from these cultivars to these genes, and none of them was found. So that means these genes are real missing during domestication process. Yeah, so the conclusion is that a large portion of disease-resistant genes have been lost during watermelon domestication. So this domestication process, which have focused on desirable fruit trait and yield. So it's not focused on disease resistance until recently. Okay, next I will talk about using this data, the resequence data we generate and other resources we generate. I show you several examples on how we identify QTLs using GWAS approach and also QTL mapping approach. So we did on the GWAS analysis on FAN1 resistance. We identified QTLs on chromosome 1 and chromosome 3, and also FAN2 resistance. We identified QTLs, a major QTL in chromosome 11. And the sugar content, we identified a major QTL on chromosome 2. And this one I will talk a little bit more later. It's a bitterness. It's a major trait during domestication. And I don't think probably no one would like to eat a bitter watermelon. So this is was mapped to the chromosome 1. Later we clone this gene. So that's using the natural accessions, the 134 natural accessions to do GWAS analysis. That proves that it can identify some interesting QTLs. So we have a recombinant inbred line population, real population. So this real population was derived by cross from this 9713, the East Asia ecotype, and also this wild watermelon. So we have whole genome sequence, de novo genome sequence for both. And we generated this real population, and we did a shallow genome resequence for this 96 reals. And we generated like 247 gig data, and the average genome coverage was about 5.2x for this real lines. We checked the recombination pattern, and also the origin of the genome from which parent, right? So this blue means that this region is from 9713. So red means this region is from that PI, from that wild watermelon. And gray means heterozygous. That confirms that this population is high quality. You didn't find any green, at least I couldn't find any green from this figure that are heterozygous, because this is a highly inbred real population. Confirm this the quality of this population. And we use this population to identify additional QTLs, or use kind of like a complementary approach for QTL mapping. And you can see this for FAN1 is confirmed. This is the peak of the FAN1. And FAN1 also mapped to the same region as using GWAS, but this one did not map this locus. But for FAN2, we identified this major QTL, also identified in this population, but we identified another QTL on chromosome, I believe it's chromosome 9. And for sugar content, it's confirmed the GWAS data is also on chromosome 2. We identified this QTL. Then we did further analysis. We mapping them to very narrow region between these two beam markers. So within this region, we found a large cluster of transmembrane sugar transporters. And when we compare to do some comparative analysis with the melon sugar QTL also contains this transmembrane sugar transporters. So we did some functional analysis, and turns out that one of them indeed can transport sugars. Okay, this is the fine QTL mapping of a bitterness trait. So we cloned this gene, and first this is the GWAS analysis. So it's first mapped to the chromosome 1. Then we use this real population and narrow down to this region. And then we use 1647 F2 population and narrow down to these two markers. So these two markers. And it's highly it's co-segregated with this indel marker. We didn't find any recombination events for this marker. And we narrowed down to, I think it's about, I forgot the region. The region contains 5 genes. And one of them, we found it's a 200 base pair mitochondrial sequence inserted at the first exon in non-betal accessions. So this 200 base pair mitochondrial sequence insertion in the non-betal accessions are highly consistent within this 96 real population and also in the natural population. So in the natural population, we checked whether they are betal or they are not betal. And for non-betal accessions, they all contain this 200 base pair mitochondrial sequence insertions. For betal watermelon, they do not contain this one. So I think this 200 base pair insertion disrupt the function of this gene, then it will not generate that betal chemical. No. Okay, here's before I go to that new project, introduced to that new project, I would like to provide a summary on what I talked so far. So firstly, is a high-quality reference genome have been generated for an East Asia genotype, or ecotype, I'm sorry, and American ecotype, and a wild Cetroides accession. So compatible genomic analysis provide novel insights into watermelon genome evolution. And the resequence of the genomes of 134 diverse watermelon accessions generate a large comprehensive watermelon genomic variation map. This provides another very useful resource. And based on this genetic variation map, we found that a cultivated watermelon has a very narrow genetic diversity. That's causing the bottleneck for watermelon breeding. And also, we found that a large portion of disease-resistant genes have been lost during watermelon domestication. And based on the sequence analysis, we found that this one, previously classified as a watermelon species, should belong to melon. And the subspecies, the Cetroides, and could be a separate species. It should not be classified as a subspecies. And the GWAS analysis identified QTLs from a number of agronomically important traits, so including the fund resistance, sugar content, fruit size, and the betal gene was further cloned through QTL fine mapping. So we are in the process of doing functional confirmation of this bitterness gene. So last, I will spend like 2 or 3 minutes talking about this KickCap project. So this is a newly funded project by USDA NIFA. It's through the specialty crop program. So the title of this project is leveraging applied genomics to increase disease resistance in cucurbita crops. This is the beginning of a new project to develop additional genomic resources for the cucurbita community. Rebecca Grumet from Michigan State is the PI of the project, and we have 21 co-PIs from 11 institutions. So I'm the leader of the genomic and the bioinformatics team, and Michael Mazurik is from plant breeding department. He is leading the squash team, and also Christian Smart also involved in this project. So we have this project has 3 objectives, the big objectives. The first one is develop genomics and bioinformatics breeding toolkits for accelerate crop improvement across the cucurbitaceae. And the second is perform genomic assisted breeding to integrate disease resistance in cucurbit cultivars. So the third one is perform economic impact analysis of cost of production and disease control, and provide readily accessible information to facilitate disease control. So I'm highlighting several points. So because these 3 big objectives have a lot of small objectives, I can't go to too much detail. So in the bioinformatics team, there are 2 main objectives. The first one is perform GBS analysis of PI, that means germplasm collections of the 4 species. We probably do GBS for about 1,500 collections for each species. That's about 6,000 species. Then we from those species, based on the GBS information, we identify a core collection of 384 accessions for each of the 4 species that can be used for the community, used as a community resource for GWAS analysis. So in total, we probably do 10,000, do genotyping for 10,000 accessions. We provide access to cucurbita genomic tools and a database via this international cucurbita genomic initiative website. This website was developed by my group about like 7 or 8 years ago, was also maintained. So this database was developed in my group. During the past like 7 or 8 years, I didn't get any funding for this database, but now finally we got some funding to do this. So we plan to make a huge improvement of this database. We did get some small money from the company, but that's not compared to this big grant. So here is a list of diseases that are the focus in this project. So this the fusarium wilt, that's a major focus for watermelon, and downy mildew is for cucumber, and also we have others, the gummy stem blight, and also the viruses. Okay, so with that, I would like to thank everyone involved in this project. This is a huge consortium, and I couldn't provide all the list in this slide, but I would like to emphasize several key people in this project. One is Hongho Sen from my group. He did most of the assembly and the genome analysis. And also Shao Gui, he was a visiting student in my lab, and now in this national NERCV. So he also did the genome analysis, and the QTL mapping and the major wet lab work was done in Yongshu's lab in this institute, and all other people contribute to this, and the funding support the work in my lab for this project was from National Science Foundation, USDA, and city companies. So I hope you have fun with watermelon.
This has been a production of Cornell University, on the web at cornell.edu.